Okay, I think we're going to make a start. Can everyone hear me and see me? If you can one of you just raise your hands, because we've been having some technical difficulties. So can everyone just, if you can raise, yeah, so you can all hear me and see me. Thank you very much. Okay, let's get started. So thank you very much to everyone that's joined this week. I hope you're all doing well. Thank you so much for the kind comments you guys have been sending regarding the webinars, and that you guys have been finding it useful. I hope you found the 2 previous sessions on photosynthesis really useful. If you have any questions, please do get in touch. You've got my Instagram and my email. If you've got any questions, please let me know and we can try and sort them out. So I know a few of you, let me just look at this. So I know a few of you are new here. So my name is Ephraim. I'm a medical student studying at Barton, London, and I've been a private tutor for over 3 years now. I know how difficult studying in lockdown can be. So I've started this free webinar to allow people that have a compromised education due to this coronavirus to have some kind of meaningful education. I can't wait for you to stay in this journey with me. We're going to go through the whole, as much of the A-level as we can possibly get through in this period, and I hope I can add some value in your revision. So I know UCAS is open, the UCAT website is now open for booking, so if you guys have any questions regarding that, if any of you are applying to medicine, please do get in touch. We can, if you've got any questions, we can discuss that, or if you have any other queries for that matter, please do get in touch. Just some few house rules. Everyone's here to learn, so please don't mess around during the lesson. If you have a question, please do raise your hand. If you have any doubt, put it in the chat. I'll be reading it regularly, and I can answer some of your questions towards the end. And most importantly, your A-levels are probably the 2 most important years, probably in your life, I'd say, because it decides your career. So stay focused in these sessions, and in general, stay focused in your revisions, and you will succeed. So this PowerPoint, as well as some other notes and resources, will be available on a shared drive, which you can all have access to if you go and follow the link on my Instagram. So if you do need to make any other notes, please do feel free. So a couple of exam revision tips. I know people like to ask me this question. Personally, I recommend staying away from doing written notes that you highlight and add to, are really time consuming to actually prepare. Personally, my favorite exam revision technique is to actually spend some time reading the content and committing it to memory and trying to actually understand the concepts behind it, which are sometimes quite challenging in A-level. If you're struggling to remember things, I recommend using Quizlet or any other flashcard app like Anki to try and commit some of those harder topics to memory. Obviously, exam papers are your best friend, so memorize those mark schemes. Even if you know every bit of content in biology, if you can't answer it like the mark scheme answers it, you're not going to get the mark. So try and get as much exposure as you can to your specifications exam papers. If you're done with your specification, go to different specification like OCR, EdXL, AQA, try their exam papers as well. They will give you good exposure to other types of questions. I really vouch for CGP and the revision guides, so I highly recommend using them. Your textbook, obviously a great resource, but a lot of it is actually unnecessary information, so use it only for wider reading. So let's get started. So I know a load of people have been getting in touch saying biological molecules have been causing them a lot of problems. So we're going to spend the next couple sessions, or as many sessions as we need, to go through the main biological molecules in A-level biology. Let's get started. So biological molecules, they're really important to understand. I know this is one of the first topics you learn in A-level, but throughout this A-level, questions will be revisiting the concepts you've learned in biological molecules. So the 4 main ones we learn about are water, carbohydrates or sugars, lipids, and proteins. Proteins are really important. I know a lot of you have ambitions to study the sciences and medicine. Proteins form some of the basis of modern disease, modern genetics. For example, Alzheimer's. Recently, it's been discovered that these tau proteins, these proteins called tau that are found in the brain, are involved in how Alzheimer's comes about. So these proteins are misfolded and start to tangle up and then start to cause problems to neurons that carry memory information. So these proteins are really important to understand. This topic in general has a lot of diagrams to them. loads and loads of diagrams, loads of reactions you should understand. So try and group them together in the different biological molecules. The 4 main ones for all the specifications are above. Try and group them together, try and spend some time drawing them out. A couple minutes every day should be fine to try and commit these to memory. Let's start with our first one. I'm going to be quite biased here, but I think water is probably the most important biological molecule. You're going to hear me saying that a lot because every biological molecule is really important. Water is basically the basis of all biological molecules. Without water, you can't transport these molecules, you can't make these molecules. So water makes about 60% of your body weight, and inside your blood, so if you took a sample of your blood and you separated the red blood cells away from that plasma, that yellowy liquid, about 90% of that blood plasma is actually water. So it's really important. In approximately 70% of the volume of most cells in the body and also plants are made up of water. So it does make up a significant amount of weight. So there's loads of functions of water, but in A-level biology, we're going to meet these examples. So first of all, water forms a stable habitat, whether it's as in a liquid form or a solid form in ice. When it's in a liquid form, it provides a habitat for fish and other floating animals. When it's frozen, the ice caps, those ice mountains, provide a habitat for polar animals such as polar bears. Water also acts as a solvent. So we're going to learn about how the characteristics and the structure of water allows it to be a solvent. But in general, a solvent is some kind of liquid that transports other molecules. For example, water can dissolve things like ions and transport the ions to wherever they need to go in the body. That's another main function. Another main function is that water can actually maintain the homeostasis in the body. So homeostasis is a massive chapter in the A2 content. But homeostasis is basically maintaining a constant internal body environment. This can be temperature, pH, the levels of ions, sugars, but when we talk about water, water is involved in the maintaining of a constant internal body temperature by a process like sweating. For example, sweating takes away body heat and thus prevents the inner temperature, the core temperature from overheating. So we're going to discuss all of this in a lot more detail, obviously. And in this whole topic, we're going to be looking at many different reactions, combining monomers into polymers and many of those kind of reactions. And in general, water is one of the key ingredients to doing any of these reactions. So last time we were talking about photosynthesis, and a lot of you know what photolysis is now. Photolysis we met in our non-cyclic photophosphorylation to regenerate the supply of ions and electrons. And photolysis is really important, and water, broken down by light, provides the reactants for photosynthesis. Also, these hydrolysis and condensation reactions, we're going to come across them a lot in this topic. Hydrolysis basically means the breaking down of a substance using water. So lysis, you're going to see a lot in A-level biology. Lysis means breaking down. So hydrolysis or hydrolysis is the breaking down with water. Condensation reactions is the forming of water as a product of joining monomers together. So for example, if I join together 2 amino acids, I get a peptide, but a dipeptide or polypeptide, but I also get water. And we're going to see where they get that from. So the structure of water. Let's go into the structure of water. So this is a really commonly asked question. Exam boards love to ask this question. So it's really important to understand whether you do chemistry or not, how to draw water. So obviously water is H2O. The way you draw it is this. That's a H2O molecule. These bonds over here, these are covalent bonds. And if you don't know what that means already, you'll learn about that later on in chemistry. If you don't do chemistry, just remember these are covalent bonds. Water has a charge to it. It's not it's a polar molecule, meaning it has some kind of charge to it. And if you don't do chemistry or you're not planning to do chemistry, don't worry about it. Just remember that hydrogen has something called a delta positive partial charge. Delta, the way to draw it is basically an incomplete 8. That's your delta. So I say hydrogen has a delta positive partial charge. Oxygen has a delta negative, and the other hydrogen has a delta positive. That's how you correctly draw an A-level version of H2O with its polarity, or those partial charges. So we're going to join 2 water molecules together. And the main bond that holds them to 2 water molecules together is a hydrogen bond. Really important to understand. A hydrogen bond is formed like this. So I'm going to draw two water molecules. Again, you must put the partial charges in. A hydrogen bond is basically, you represent it as a dotted line from a delta negative oxygen and a delta positive hydrogen. Really important. It's between a delta negative oxygen and a delta positive hydrogen. Really important. And then what you do is label it using an arrow saying that's your hydrogen bond. Again, if you're interested, this is your covalent bond. So don't get too confused. And many of the properties I've just talked about and that we will talk about in a few minutes all revolve around this hydrogen bond and how strong it is. Okay, so in chemistry, you learn, for you chemists out there, the hydrogen bond is probably not one of the strongest bonds. It's actually one of the weaker bonds out of the ones you learn about. But in biology, in a biological context, hydrogen bonds are extremely strong and provides the connection between water molecules as a liquid and an ice, and we'll talk about that in a minute. So I hope everyone understood the structure of water. So key properties of water. Again, this is one of those things that you have to just memorize, okay? This is a really commonly asked questions in all the examples, so just memorize it. So obviously we talked about how water forms these really strong hydrogen bonds with each other. And these hydrogen bonds, to actually break them apart, we need to put a loss of heat energy in. So hydrogen bonds form spontaneously because they're basically acting like a magnet between a delta positive hydrogen and delta negative oxygen. But to break them apart requires a lot of energy. And that energy, a lot of energy required to break these hydrogen bonds presents these following properties. So first of all, water has something called a high specific heat capacity. High specific heat capacity, or specific heat capacity, is basically, the definition is the amount of energy needs to raise 1 gram of water by 1 degree Celsius. And since in water, the energy required to raise 1 gram of water by 1 degree Celsius is so high due to these hydrogen bonds, it makes habitats very stable. So large bodies of water like oceans and rivers, they don't just evaporate on a hot summer's day. They actually stay pretty much at constant temperature regardless of the atmospheric temperature. And that's because a lot of energy is required to raise 1 gram of water by 1 degree Celsius. Hence, we say it has a high specific heat capacity. Following on from that point, high latent heat evaporation. This is basically the amount of energy required, again, to break hydrogen bonds between liquid water. This is really useful when we're talking about homeostasis in the body. So, for example, you're really hot, your hypothalamus detects internal body temperature, as well as you've got some kind of surface skin temperature receptors, but they all feed into this part of the brain called the hypothalamus. The hypothalamus detects that there's an increase in body temperature and does several things to try and reverse that. One of them is to stimulate sweat production from sweat glands. The purpose of releasing sweat is actually to put a layer of water on top of your skin. And because so much energy is required to break those hydrogen bonds, the water that's now being placed on your hot skin will use the heat energy from your skin to evaporate itself off. So by that, you're actually going to reduce the temperature of your skin and actually reduce your core temperature by using all that energy to evaporate the water off. That's why sweating is so useful. And we call this the cooling effect. Next, the next couple come together in a pair. So cohesion and adhesion. It basically describes how water molecules can stick to each other and stick to other surfaces. So water, as we know, forms hydrogen bonds. It can form hydrogen bonds between other water molecules and also the wall of a xylem. The xylem is a vascular vessel found in the plants that moves water from the roots up to the leaves in a one-way flow via something called a transpiration stream. And the way water is moved up from the roots all the way to the top of the leaves is via a process called transpiration. And it uses the water's ability to bind onto xylem walls via cohesion and adhesion, and allows it to suck it up via these really strong hydrogen bonds attaching it to the xylem wall. So that's cohesion and adhesion. Solvent, we already talked about, is the ability to dissolve substances needed for an animal or a plant. Let's draw what being a solvent looks like. So for example, we have a water molecule. You've got your H2O, and we can put our partial charges in. For any substance to dissolve successfully in water, they need to split apart and bind onto these partial charges. For example, when you add salt or sugar into hot water, you see it dissolve and becomes colourless. Let's explore why. Salt is also known as sodium chloride or NaCl. Salt is made up of two ions, Na plus and Cl minus. When salt is added to water, because these hydrogen bonds or these partial charges have such a strong influence, they start to attract these ions. So the delta positive hydrogen attracts the chloride ion, and our delta negative oxygen attracts our positive sodium ion. That salt molecule has now successfully dissolved in water, and that's what dissolving looks like. This is useful in both the xylem and the phloem in plants, as well as in the blood. For example, we need to transport glucose and ions throughout our blood to all our extremities. This is done via them dissolving in water, being able to be transported much easier. The next point is water, when it's frozen, forms ice, obviously, at 0 degrees Celsius or minus 273 Kelvin. And ice has a very special structure. So a liquid water molecule we've already seen is basically your H2O with a hydrogen bond to another H2O. You get this kind of bond. That's in liquid. In solid water or ice, water actually forms something called an open lattice structure, where loads of water molecules form kind of a ring and form hydrogen bonds in this ring. And this is now called a lattice. And this happens between loads of water molecules. So it forms a bit like a cube, but there's loads of them. So there's a lot of space. So this is all space, free space in the middle of this lattice structure. That free space reduces the density of ice. It makes it lighter than water, or less dense than water. So ice, forming this open lattice structure, is now less dense than now liquid water, so that it can float. And ice being able to float is a really important property, especially in the animal kingdom, in the polar caps, where these large polar bodies, or these ice bodies, provide a habitat for things like for animals like polar bears and seals. So ice is really important. And it's all coming down to how hydrogen bonds form this open lattice structure that's got a reduced density than liquid water, so that it can float. Lastly, it's not really in your spec, but something that's really interesting. So you've probably seen these pond skaters, these insects that basically skate or float on top of water. This is all done, so if you can see over here, they are basically floating on water. This is all because water forms so many hydrogen bonds with each other that has something called a high surface tension, so that it is not very easily deformed, so that this pond skater can now float on it. That's really interesting to understand. So the things to remember for your specification are these points. They love to ask these. So if you ever see a question saying, what are the main structures or properties of water? That's what you use, and you describe it in terms of hydrogen bonds and how much energy they require to break. Okay, we're going to skip over this point because I know some of you are actually not in year 12 yet, so this is a quite complex concept to understand, but we will definitely go over this later. It's really interesting. That's our first biological molecule done. So well done. We've done 1 out of the 4 biological molecules. Our next one, again, so important. Its function in both plants and animals is really important molecule, carbohydrates. Carbohydrates is basically a broad umbrella term for all sugars. So we've learned about photosynthesis. Photosynthesis was the forming of glucose. Respiration is basically using that glucose to make energy in the form of ATP, or adenosine triphosphate. And if you're not familiar with ATP, we're going to go through it in a few slides' time. But yeah, the majority of energy comes from glucose. When we respire glucose, we break it down to form ATP. So carbohydrates, there's a couple ways to classify carbohydrates. It can either be a polysaccharide or a monosaccharide. So polysaccharide is just a fancy way of saying a polymer. A polymer is just several monomers stuck together. And monosaccharide is just a fancy way of saying monomers. It's just a special way of saying it in terms of carbohydrates. The main hexose monosaccharide that we learned about is glucose. By hexose, I mean it's 6 carbons. It has 6 carbons to it. So for example, a pentose monosaccharide, for example, ribose or deoxyribose, they only have 5 carbons. So we call it a pentose monosaccharide. It's really important to understand that carbon forms 4 bonds or 4 covalent bonds with other molecules or other atoms. And if carbon doesn't form these 4 bonds, it's not a complete structure. So make sure when you check your diagrams, carbon should always have 4 bonds attached to other elements. So we're going to look at different biological molecules, but it's worth understanding the elements that are making up carbohydrates. And I've seen some of these questions before. So remember, carbohydrates are only made from 3 elements: carbon, hydrogen, and oxygen. Remember that. CHO, that makes up carbohydrates. No other element should be present in a carbohydrate besides CHNO. What exam board is this for? One of the Q&As. This is for all exam boards. So all exam boards will go over this in your A-level. Okay, so we've understood what carbohydrates are. Carbohydrates, I like to put some application style scenarios in so that you can answer these application style questions in your A-level. Carbohydrates like glucose. Glucose is very valuable. We know it's broken down to make ATP. But we don't store large quantities of glucose in its glucose monosaccharide form. The reason is because glucose has its own partial charge and it can actually be very damaging to other substances. Glucose acts as something called an oxidizing agent. So oxidation is the loss of electrons. So what glucose does, if you don't store it properly, is it starts to rip off electrons from other molecules, thus acting as an oxidizing agent. This is very damaging to the organism and can form many different conditions. Also glucose, too much glucose in the blood. Obviously we talked about how they can act as an oxidizing agent by ripping electrons off other structures. Glucose can do these to really important structures that maintain blood pressure and monitor blood pressure, and also cells in the pancreas that control blood glucose. And if you have no regulation of glucose in the blood, this condition is called diabetes. Okay, and we're going to learn about diabetes a bit later on. So drawing glucose is really important, and I've seen so many students that I teach not being able to draw 2 isomers of glucose. The word isomer basically means a different form of glucose. So we learned about 2 major forms of glucose, monosaccharides, these monosaccharide glucose molecules. It can either be alpha glucose or beta glucose. They basically have very similar structures besides one very important difference, but they have the same function, which is to store energy ready for use in respiration to release ATP. So, a very important concept to understand in general, if you're doing chemistry or not, is that bonds contain energy. Bonds contain energy. For example, let's consider this C to C bond, this carbon to carbon bond. The energy is stored in that bond. Energy is stored in that bond. When I break that bond, I'm going to release energy, okay? And that energy is used to go on to make ATP. So we talked about ATP in a few lessons ago, and I told you the structure of ATP looks a bit like this. And if this doesn't make sense to you, we're going to go over this a bit later on. But the energy that's stored in ATP is stored in that bond. And when that bond is broken, it releases loads and loads of energy. The breaking of a bond to release energy is called an exothermic reaction. It releases energy. To form bonds, to make bonds together, that's an endothermic reaction, and that requires energy. So, everyone get your pen and paper out. We're going to start to draw glucose. Really, really important to understand. I've seen 2 markers, 3 markers on how just to draw alpha and beta glucose. So, I have a 4-step method to draw glucose. 4-step method. The first step is something called drawing the carbon skeleton. The carbon skeleton. By this, I mean what I'm about to draw. So in both alpha and beta glucose, they both have the same carbon skeleton. We know that glucose is a hexose monosaccharide, so it has 6 carbons. So let's draw these 6 carbons. So we have a carbon to an oxygen. This is your carbon skeleton. And just note there's an oxygen there, and you'll understand why. So carbon number 1's there, carbon number 2, 3, 4, and 5. We've only got 5 carbons. We need to get one more carbon. And this comes on to my second step. This is adding the CH2OH group. So all you have to do, the next step, all you have to do for either alpha and beta glucose is just add CH2OH on carbon number 5. There we get our 6th carbon. Third step is the position or orientation of the OH groups or hydroxyl groups. Okay, so if you're drawing alpha glucose, this is the main difference between alpha and beta glucose, the position and orientation of these hydroxyl or OH groups. And the way I remember it is using this kind of silly way to remember it. So all you have to say to yourself is down, down, up, down. Just remember down, down, up, down. Down, down, up, down refers to the orientation of the hydroxyl groups in alpha glucose. So that's carbon number 1, carbon number 2, carbon number 3, and 4. So on carbon number 1, the orientation is facing downwards. We'll draw it downwards. Carbon number 2 is also down. Carbon number 3, up. Carbon number 4, down. That's where that down, down, up, down comes from. For beta glucose, the main difference between beta and alpha glucose, a very commonly asked question is, on carbon number 1, so this one over here, the hydroxyl group is flipped by 180 degrees. This way we're forming 2 isomers or 2 versions of glucose, and they have different functions to form different polysaccharides. But the way to remember it is just flip carbon number 1. So if you remember down, down, up, down, you just flip the first one, make it up, down, up, down. So let's draw alpha glucose. This is your alpha. The 4th step is, remember that fact I told you that carbons require 4 bonds? Let's see how many each bonds this carbon has. This carbon has 1 over here, 2 over there, and 3 over there. This one only has 3, and they all have 3. All the carbons only have 3 bonds here. So we need to add some bonds to make it 4. And we do that using the only other elements found in carbohydrates, which is hydrogen. The last step is add hydrogen atoms. So you just add, wherever there's not 4 bonds, you add a hydrogen. And the one everyone forgets is this one on carbon number 5, this one over here, add a hydrogen there. That's your complete alpha glucose. So to draw beta glucose, the only main difference, remember, is the orientation of the hydroxyl group on carbon number 1. So what I'm going to do is going to flip it around. So it's up, down, up, down. And there you have your beta glucose. Okay, that's your beta glucose now. So remember the 4 steps. Carbon skeleton. We'll do this one more time. Let's draw beta glucose. The first step, the carbon skeleton, is this. So the same for both. Second step, add the CH2OH group. The third step, positions and orientations of hydroxyl. So this is beta glucose, so it's now up, down, up, down. Fourth step, add hydrogens. And there you go, in about 20 seconds I've already drawn beta glucose. That's how you do glucose. So now you guys know how to draw alpha and beta glucose. So there's a couple more different monosaccharides we need to learn about. And the 2 other most important ones you should know is ribose and deoxyribose. So we know that glucose is something called a hexose monosaccharide. It has 6 carbons. Ribose and deoxyribose only have 5 carbons. And hence we call them a pentose sugar. You'll come across ribose and deoxyribose when you do nucleotides probably next time. Nucleotides are examples of nucleotides would be RNA, DNA, ATP, ADP. These are really important nucleotides, and they make up things called nucleic acids or polynucleotides. And this is how DNA is made. So you need to know how to draw this as well. And it follows some of the same steps as before. So the first step is your carbon skeleton. This is different because there's only 5 carbons, and it looks a bit like this. So for both deoxyribose and ribose, it looks like this. That's your carbon skeleton. The second step, remember, the CH2OH group. This time we add it over here, always onto the last carbon in the skeleton. The third step, the position of the hydroxyl groups. In ribose, the way to remember is up, down, down. There's only 3, so up, down, down. That's carbon number 1, carbon number 2, and 3. So 1, 2, 3, 4, 5. So up, down, down. The fourth step, add hydrogen, wherever there's not 4 bonds. So 1 there, 1 there, 1 there, 1 there. And that's it. That's your ribose. So the main difference between ribose and deoxyribose is this deoxy thing over here. That's referring to on carbon number 2. So carbon number 2 is this thing over here. There is no oxygen on carbon number 2, so we call it deoxyribose. Let's get rid of the oxygen. Let's get rid of the oxygen, and then we just put the hydrogen back. That's now deoxyribose. Okay, so ribose, deoxyribose. Ribose is found in RNA, in ATP, ADP. Deoxyribose only found in DNA. So DNA stands for deoxyribonucleic acid. Okay, now you guys know how to draw ribose and deoxyribose, but you're actually getting something. So now let's learn how to join monosaccharides together. Okay, and we talked about it at the beginning, to join any monomer in biological molecules, not just monosaccharides, any monomer. To join any 2 monomers together, it's a condensation reaction. It releases water. And to break apart any polymer or dimer, that's a hydrolysis reaction. You put in water to break it, to break those bonds. So the main bond found in monosaccharides is something called a glycosidic bond. This is the bond found between monosaccharides. And glycosidic bonds are covalent bonds. So we looked at hydrogen bonds, these are covalent bonds. So the way to classify carbohydrates, if it's just one monomer or a monosaccharide like ribose, deoxyribose, alpha, beta glucose, you call it a monosaccharide. If you join 2 monosaccharides together, only 2, we call it a disaccharide. And 3 or more monosaccharides joined together, it's called a polysaccharide. So remember that glycosidic bond. Let's draw this glycosidic bond now. Okay. So the most basic disaccharide that you should be able to draw is something called maltose. Okay, maltose is a disaccharide, and it's made up of 2 alpha glucose molecules. Again, a really commonly asked question is 2 alpha glucose molecules joined together using something called a 1,4 glycosidic bond. Okay, we'll understand where that 1 and that 4 comes from. So let's draw maltose. So first of all, draw both your alpha glucose molecules together, side by side. Okay. Now let's get those hydroxyl groups in. Well, let's first put the CH2 groups, CH2OH groups in. Then that hydroxyl group, so this is 2 alpha glucose, so it's down, down, up, down. So down, down, up, down, and likewise over here, down, down, up, down. The last step was just add hydrogens everywhere. Again, a really commonly asked question is how do you draw maltose and what's it made up of? 2 alpha glucose molecules. So it's joined by a 1,4 glycosidic bond. So glycosidic bond is a general term for any bond found between carbohydrates. The reason it's called 1,4, let's label the positions of these carbons. This is carbon number 1, 2, 3, 4, 5, 6. Same over here, 1, 2, 3, 4, 5, 6. We're going to join 1 and 4 together. Hence, we call it a 1,4 glycosidic. That 1 to 4 is the positions of the carbons. We know this is a condensation reaction, so we need to form water. So where do we get this water from? H2O. So just that's your H2O molecule. So we're going to release H2O. So we're going to remove that H2O from those 2 hydroxyl groups on carbon number 1 and 4. And we're going to form our glycosidic bond, which looks a bit like this. That's a glycosidic bond. Okay, and what you've released is the water from those 2 hydroxyl groups. You've now released water. Hence, this is a condensation reaction. So maltose is just 2 alpha glucoses put together, a disaccharide. Amylose is a component of starch. So starch, again, we're going to learn about this in a few minutes' time, but starch is the main storage of glucose in plants. Okay, starch is made up of 2 components, amylose and amylopectin, and they have different structures. Okay, both amylose and amylopectin are only made up of alpha glucose molecules. So starch is only made up of alpha glucose. So amylose is basically 3 or more alpha glucose monosaccharides put together. So maltose is just 2, it's a disaccharide. Amylose is just that polysaccharide with just alpha glucose molecules stuck together using those 1,4 glycosidic bonds. The structure of amylose and amylopectin varies a bit, and that's to do with their roles in the formation of starch. And we're going to look at that next. So just remember that amylose is a polysaccharide made up of 3 or more alpha glucose monosaccharides. And maltose is just 2, so it's a disaccharide. Does anyone have any questions, or should we keep going? So if anyone has any questions, put it down on the Q&A, otherwise we'll just keep going. So we've learned about maltose. Maltose is a disaccharide. Okay, you don't need to know how to draw any of these disaccharides except maltose. Okay, but other disaccharides you need to know the structure of, or the monosaccharides that make up these disaccharides, are these 3 over here: sucrose, maltose, and lactose. So we know maltose. Maltose is 2 alpha glucose molecules stuck together using a 1,4 glycosidic bond. Sucrose is another really important sugar in the human body. It's formed by alpha glucose and fructose. Fructose is a different type of monosaccharide. Just like alpha and beta glucose, fructose is a different monosaccharide, which you don't need to know its structure. Just remember, sucrose is made up of alpha glucose and fructose put together to form a disaccharide. Lactose is made up of 2 different monosaccharides. One's called beta-galactose. Again, just remember this. And either alpha or beta glucose put together with a beta-galactose forms lactose. Again, a disaccharide using 1,4 glycosidic bonds. So again, just some factual information you need to remember. So types of glycosidic bonds. We've looked at 1,4 glycosidic bonds. Okay, we've looked at 1,4. 1,4 glycosidic bonds form straight chain polymers. Straight chain polymers, or dimers, or like disaccharides, basically. So if you ever use a 1,4 glycosidic bond, you always form straight chains. It just goes on and on and on, stuck together in a straight chain. Eventually, that straight chain, because it's so long, will start to coil up. It will start to coil up like this. Again, it's all just straight chains. That's the structure of amylose. Amylose has a helical structure, and it's only made up of 1,4 glycosidic bonds and alpha glucose molecules. So remember, 1,4 glycosidic bonds always form straight chain polymers. The second type of glycosidic bond is called a 1,6 glycosidic bond. And again, this forms between carbon number 1 on one monomer and carbon number 6, the one on top with that CH2OH group. It forms a bond between those 2. A 1,6 glycosidic bond forms perpendicular or branched polymers. So there's 2 branched polymers we look at. So we know that starch is made of amylose, a straight chain polymer. It's also made up of something called amylopectin. Amylopectin is a branched polymer, and it looks a bit like this. So all the straight chains, for example, this straight chain, that's all 1,4 glycosidic bonds. The minute I form a perpendicular strand, for example here, that joining over here, that's a 1,6 glycosidic bond. That's the structure of amylopectin. It's also the same structure is the same in something called glycogen. Glycogen is the equivalent of starch only found in humans, and it's a storage molecule of sugar. Okay, so remember, 1,4 glycosidics are straight chain polymers. 1,6 glycosidics are branched polymers, for example, amylopectin and glycogen. So these are some polysaccharides that you should be aware of. So starch, again, is made up of amylose, amylopectin, and it's only found in plants. Glycogen is a storage molecule of glucose found in the liver and muscles. And cellulose is a plant-based polysaccharide, and it basically makes up plant cell walls. And we can't digest cellulose. So yeah, we can't digest cellulose because we don't have the enzymes which are called cellulase to break down cellulose polysaccharides. So starch is really important, commonly asked again. Starch is basically a storage molecule, so it stores excess glucose as starch. We know why we don't store glucose as glucose itself, because it starts to act as an oxidizing agent and it affects the solubility in water. So to save all that hassle, we just store it as starch. So starch is stored in these organelles called amyloplasts. Amyloplasts are basically little starch granules found inside a cell. They're found inside a cell, and they're also things called tubers. So potatoes is just one massive amyloplast, or a massive store of starch for the plant. So starch is stored in two different ways, okay? And this is to optimize its storage capacity, the amount of space it takes up, and also how available that starch is, so we can break down starch back into glucose ready to use in respiration. So it's stored as amylose and amylopectin. This point is really important. Glucose is stored as starch because starch is insoluble. It does not dissolve in water, so it doesn't contribute to something called osmolarity. So I didn't talk about osmolarity a lot because I know some of you haven't covered this yet. But osmolarity just refers to how polar molecules can attract water by a partially permeable membrane. So for example, if you consider a red blood cell, if that red blood cell is packed full of glucose, so just loads and loads of glucose, because glucose is in such a high concentration here, it means that water is in a low concentration in that area. So water starts to flow into the red blood cell, causing it to swell and eventually burst if it's not controlled. That's osmolarity in a very basic way. But the main reason why we store glucose as starch is because it's insoluble, so it won't contribute to that osmolarity effect. So how is cellulose different to callose? Okay, good question. So if you guys don't know what callose is already, you'll learn about this in plant immunity. Okay, so cellulose is a straight chain polysaccharide. It's a polysaccharide, and it makes up cell walls. Callose, so if I draw a plant cell here, callose is basically a different form of a polysaccharide, but it's used in an immune function, not to make up cell walls. So what happens is, for example, if a plant is attacked by a pathogen, the first line defense for a plant is to do something called callose deposition. So callose is a really strong, thick polysaccharide. What the plant does is it synthesizes loads of callose and sticks it in between the cell membrane and the cell wall and prevents things getting inside, prevents the pathogens entering the cells. This is called callose, and the process is called callose deposition, and we'll learn about this a bit later on. That's the difference between callose and cellulose. I hope that answers that, your question. So amylose and amylopectin, again, structure we'll talk about one more time. Amylose structure is a coiled helical structure. Amylopectin is a branched polymer. This is for two different reasons. Okay, the reason amylose is so good at storing glucose is because it only has two free ends. These are free ends over here. And there's only two of them, so they can coil really close together. It can compact so tightly and store so much glucose that it's ready to use if the plant does need it. What happens is, to prevent this coil unraveling and prevent all this really tightly bundled amylose uncoiling, we form hydrogen bonds between glucose monosaccharides, and this holds together the helix. So amylose, the positive points are it's compact, and that's mainly, and it conserves space in the plant because it's so compact. That's the two main reasons that amylose is so good at its job. Secondly, we have amylopectin. Amylopectin is a branched structure. The reason that it's branched is because it has so many free ends. These are called free ends, where they're chain ends. There are so many of them. So in amylose, only two. In amylopectin, there's loads of them. When a plant wants to access this store of amylopectin, it has to break down these free ends using an enzyme and work its way down the chain and release all the glucose. So just release loads of glucose from those free ends. The more free ends you have, the more enzymes can attach. So enzymes attach onto all these free ends, so one over there, and start to rapidly break down these polymer chains to access the glucose. So if there's loads of these free ends, you can access glucose really quickly. And that's not the case in amylose, because amylose is designed to be very compact and store large quantities in a very small space. Amylopectin, its positive points are it's readily accessible due to branching and many free ends. Hope that makes sense. So you can release loads of glucose in a short space of time because amylopectin has so many free ends that enzymes can attach onto. Good. One last thing I wanted to mention. So amylose obviously is a straight chain polymer. So any straight chain polymer uses 1,4 glycosidic bonds. Since amylopectin is a branched polymer, any branching that's perpendicular, so if you want to form a straight chain, it's just 1,4 glycosidics. Loads of glucose attached by 1,4 glycosidics. The minute you want to branch something, or branch again, and this resembles the structure of amylopectin, in that branch place, you need a 1,6 glycosidic bond. And if you look at the carbon number 1 and carbon number 6, you can understand why a 1,6 glycosidic bond is really useful. Don't worry, you don't need to know how to draw a 1,6 glycosidic, you just need to know how to draw a 1,4. But you should be aware of what the function of a 1,6 glycosidic bond is and its main function in amylopectin. It's also found in a structure called glycogen. So glycogen is the human equivalent of starch. It's the human equivalent of starch. Let me just check if we have any questions. Okay, good. So yeah, glycogen is basically used by animals as a long-term storage of glucose. We know why it's not stored as glucose, because it acts as an oxidizing agent, and it also contributes to osmolarity. So glycogen is that storage molecule. It stores glucose in glycogen in the liver and in pretty much all muscles in the body. And the structure, again, resembles amylopectin very much, except you have more branches than before, so that there are so many free ends that loads of enzymes can break down glycogen very quickly to act as a really quick source of glucose for the animal. This is very compact, and it contains both 1,4 and 1,6 glycosidic bonds. For those of you that are interested, the enzyme that breaks down glycogen using those free ends, the enzyme that does that is something called glycogen phosphorylase. That's the enzyme that breaks down glycogen to release those glucose really quickly. Glycogen phosphorylase is controlled by those hormones insulin and glucagon. Again, we'll talk about that later. And any dysfunction in how insulin and glucagon works contributes to diabetes. So what would you, so I've told you that glycogen phosphorylase is the enzyme that breaks down these free ends and releases the glucose. There's a condition called, there's two conditions actually. There's one called McArdle's syndrome and Her's syndrome. These are two diseases where in the person's gene to code for glycogen phosphorylase, that enzyme, that gene is not present. So they cannot form any glycogen phosphorylase enzymes. So people with these conditions, McArdle's and Her's syndrome, they can't readily use glycogen stores. So they actually get symptoms of diabetes. They can't control their blood sugar. They get tired really quickly. And their muscles start to cramp, because we know that glycogen is stored in muscles, but if it's not accessible for active or respiring muscles, they start to cramp, and just something called lactic acid metabolism, and we'll learn about that in respiration. That's the reason why these people with McArdle's and Her's have these muscle cramps and fatigue, symptoms of diabetes, because they're not having this enzyme called glycogen phosphorylase. So let's end on cellulose. Cellulose, again, it's the main polysaccharide that makes up cell walls of plant cells. It does not make up the cell wall of bacteria. And that's the main differences between eukaryotic cells and prokaryotic cells. In eukaryotic cells, all cell walls are made up using cellulose. In prokaryotic cells, all cell walls are made up of something called lipopolysaccharides. And if you're an EDIC cell, you should be able to understand how gram staining works. And if you're not too sure about that, let me know and we can discuss it later on. So cellulose is a straight chain, unbranched beta-glucose monosaccharide joined together with 1,4 glycosidic bonds. Okay, and it forms a straight chain polysaccharide because of these 1,4 glycosidic bonds. Remember, cellulose has beta-glucose, not alpha-glucose. Okay, so this is the structure. This diagram here resembles the structure of cellulose. So these structures here, these are your long polysaccharide strands of cellulose. We call these microfibrils. Microfibrils are joined and held together by hydrogen bonds, and it keeps all those strands together. So several microfibrils put together with their hydrogen bonds makes up something called a macrofibril. Loads of macrofibrils make up fibers, and loads of fibers makes up these cellulose cell walls. And again, they're only used in plant cell walls. The key things to remember: beta-glucose, 1,4 glycosidic, straight chains, microfibrils, and they're held together using hydrogen bonds. That's all of your water and your carbohydrates done. Okay, so the next two biological molecules we'll do next time are your lipids and your proteins. Proteins is quite a long process, but it's a really, really, really important biological molecule, and the structures it makes are really important. And it's very useful to know, and it's also very important to understand protein synthesis, and that's another topic that we'll cover at the end of this. So that brings me on to the end of our third session. Just a couple of things we need to add. So let me just get up PowerPoint. So I hope everyone found that useful. Again, if you have any questions, do let me know. This PowerPoint is available on the Google Drive. Just a couple of things I need to add. So these free webinars will always remain free, okay, for the first 100 people that join. Again, the Zoom link will always be the same, and it'll be on my Instagram bio. Please follow me on Instagram. I do post loads of updates on tips for personal statements, tips for biology, and loads more. So definitely do give me a follow. However, due to time constraints and other constraints, we can't cover all the topics in A-level, especially because there's differences between the different exam boards. So I know a lot of you are entering year 13. So my focus will be year 13 content, unless something like biological molecules, which was highly requested. That's an exception. I don't think we'll be able to cover a lot of year 12 content. So we'll only be covering common topics, for example, the kidneys, the nervous system, muscle contraction, anything, those differences between the specifications I won't be able to cover. For example, if you're on EDIC cell, you need to know how to do gram staining, and gram staining is a large topic that you should understand. So be aware that does leave out some topics which I'll make you aware of. However, I run a mentorship scheme. So I've been a private tutor for over 3 years now. If you want to get private tuition from me, I offer a 1 hour free, not free lessons, it's a 1 hour lesson per week, worth up to £20. I offer you past paper questions from really exclusive resources that teachers have access to. You'll get quarterly mock exams, really helpful notes, past paper questions. You'll also have access to ask me any questions during the week you have on your application to university, and if you're doing medicine, even better. I'll also be giving you personal advice on your application to medicine, so interview tips, work experience contacts, those kind of things. And also one edition of your personal statement will be marked before the UCAS deadline. That's another service I offer, which is £30 bought separately. However, if you want all of that, that's only £15 a week. So for £15 a week, you get all of that. For more information on that, please go to my Instagram. You can see, or DM me. There was only, we started with 7 slots. We've had, last week we had 3 slots remaining. Unfortunately, there's only 1 left. So if you're really wanting this mentorship scheme, there's only 1 slot left. So please do get in touch if you're interested. If you're interested in editing your personal statement, I offer a really comprehensive personal statement service. So I discuss your CV beforehand. We discuss what you can add, what you should remove. We analyse some successful personal statements. One edition will be personally hand-marked and critiqued by 2 medical students who've got 3 or more offers to medical school. And then we'll debrief these edits and suggest any improvements on another call. That's £30. So do get in touch if you're interested in that. So thank you very much for listening to this 3rd webinar on biological molecules. We'll be here every Thursday at 5 p.m. So if you're interested, please do join. Any updates, please email me. Any questions, DM me on Instagram. But otherwise, if anyone has you're more than welcome to ask any questions. I'll be around for a few more minutes. So thank you very much. This whole webinar will be recorded and placed on YouTube, as well as all other previous webinars and future webinars. So do watch it if you need extra help. So thank you very much, everyone. I'll see you next week, Thursday at 5 p.m. If you have any questions, I'll be sticking around for a few more minutes to answer them. Thank you. Someone's asking, do we need to know how to draw cellulose? You just need to remember that it's a beta glucose, it's made of beta glucose, and it's a straight chain polymer with 1,4-glycosidics. I don't think you need to know how to draw this because it's quite time consuming. Just be aware of it. I did put a picture on the PowerPoint, so do be aware of what it looks like. Remember, it has an inverted 180-degree structure. Because beta glucose has a different orientation of hydroxyl groups. Just be aware of what it looks like in case. Will we go through exam papers in the presentation? Unfortunately, that's one of the things that we don't have time for. If you're interested in that, I do have access to exclusive questions from the exam boards. So that's offered in my mentorship scheme, my private tuition service. So if that's something you're interested in, do get in touch.